diagnose a case is always put the microscopy or EM. There's a technique. Okay, there's a technique to discuss now because there are so many branches of microscopy. Okay, but we're going to focus on a few things on the optical, the electron, and the scanning microscopy. Okay, so the reason why we have to use microscope because you know that the biological material that we're going to observe and the agent we're going to observe are very, very small, and they are not like parasites, okay, like insect that you can see by just using a stereo microscope. In our case, we're going to use a higher definition microscope. At least it has to be enlarged about 1,000 times, okay, for us for able to see the changes or the agent itself. Okay, for virology, there are so many procedures out there. Okay, but there are two general procedures that we're going to focus mainly for detection of EM, namely the negative staining, EM, or the thin section electron microscopy. Okay, here when we do diagnosis by EM, by the short word of the full name is electron microscopy, we are actually directly viewing the virus morphology, okay? And we're going to determine by looking at the morphology, we are able to determine the virus family because virus family, regardless of their genera, their strain, they have the similar feature. They don't change at all. They still have the capsid, they still have the envelope, or if they are spherical or they are elongated, they are all similar. It's just that you're not able to differentiate from which are their genera, okay? And for some viruses, okay, they are named after their characteristic features, okay, or the surface distinctive, okay? So what's the advantages of EM, okay? For some disease diagnosis, EM is very useful when we are working with a virus that is not able to be cultured. Okay, there are some viruses outside there causing a disease, but when we take the sample and put into a cell culture, they just do not able to induce CPE. So we're not able to see what are the changes they induce, but we can easily observe them under the electron microscopy. Okay, and of course, as I mentioned just now, by looking at the CPE sample, after processing the sample, concentrate the virus, we can look at the virus, we can look at the morphology, and from there we can easily diagnose at least which family they come from. Okay, for your information, if some of you remember in one of your lectures with one of the virologists, you're talking about Nipah virus. Okay, Nipah virus. Let me tell this on. Nipah virus is caused by paramyxovirus virus. And Nipah virus is a virus got its name from a place called Nipah, in which the virus was being isolated. Okay, Nipah virus. Okay, this is isolated in Malaysia. Okay, Nipah virus. Okay. And first Nipah virus outbreak. Okay, in our pig population. Okay, nobody knows. Okay, in pig population, nobody knows what is the cause of CNS in the pig. Okay, so what happens that we take a sample from the pigs, okay, of course the saliva, the brain, and we put in a cell culture. In the cell culture, at that time, people always think this is due to JEV. You know, JEV, another virus that causes a CNS problem, but nobody know that this is a newly emergent virus. Okay, people don't know. People always thought it's JE. So we actually on that time we misdiagnosed it with JE, and there's a lot of problem because we realized that it's not JEV because after giving a vaccination to the pig with JEV, the animal and the farmer succumb to the disease. Okay. So what we do on that time, after taking a sample from the pig, we put in a cell culture, we are CPE, and then we observe under EM. Okay. Because we want to know which family of this virus. Okay, by looking at the MAM, then we find out this virus have this characteristic, and that's how we know that this is due to paramyxovirus. Okay, paramyxovirus, if you remember your class, is a virus that is pleomorphic, got envelope, got spike. Okay, and that's how we diagnose that actually Nipah virus, okay, belongs to the paramyxoviridae, and it got the name of Nipah because there's a place where it was isolated in 1996, if I'm not mistaken. And after 1996, okay, the virus has spread to many parts of the world. Okay, and it's believed the virus is being transmitted by bats because people find that it's not actually accessible pig because in country like certain country they don't have pigs but they have the Nipah virus among the population. Okay, so this is a bit about the important or the significance of electron microscopy in doing a diagnosis on these two category of viruses, non-culturable virus or virus that we don't know. Okay, and we just have to find the virus family. Okay, so following that, after we find that this virus is virus, then only we do the, you remember in my class, we are talking the other day, we are talking about, we do serology. Okay, serology test. Serology test is a test that we testing of this our antigen with several type of antibodies belong to the paramyxo family. Okay, from here we know that when this antigen is not been neutralized with this antibody, we know that this is not a similar serotype. This is going to be a new serotype. For example, if we tested against Hendra virus, okay? Hendra virus is also a virus of the same family, but it is originated from the horse. Okay, so after doing the serology test, we find that this so-called Nipah virus not able to be neutralized with this virus, and from there we know that this is a new emergent virus which is not here before, okay, on that time, but now it's everywhere in many parts of the world. Okay, what's the advantage of this EM? Okay, the advantage is that, as I mentioned just now, you can only see the morphology, but you won't know whether they are different variant, what are the strain, is it a recombination, we won't know, okay, because it's just a morphology. And a person that is going to examine the EM has to be a specialized or skilled person, have to go a lot of training in order to operate the machine, okay. And in order for us to observe the copy number of the virus, it requires a very high copy number of virus. That is 10 power of 6 virion, meaning that our sample had to be really concentrate. If we got only few hundred particle, it's not enough. Okay, so I would say that this technique is less sensitive. Okay, it's specific but less sensitive. Okay. Okay, this is a very general, the advantage of electron microscope. Okay, first of all, the advantage, if you have a sample, you can magnify, okay, 500 times. Okay, you can observe or 2D or 3D, and you can also observe the surface of the cell with a scanning microscope, scanning electron microscopy. Okay, this advantage, the equipment is not small, it's very big, it occupies a big room, very expensive, and in some cases, the specimen need to be kept in frozen. Okay, this is some of the disadvantage. And for information, not many institution has the electron microscopy. Only a few, and we are lucky. UPM we have this in these facilities, and it's situated in Institute Bioscience UPM. Okay, so we can use the facilities to diagnose cases of EM. Okay, so EM is considered one of the direct detection method whereby you have a sample. For example, animal come up with diarrhea, you can take the sample, you just pelleted the sample, and then you can choose examine the sample under the microscope and can examine for the specimen. Okay, this is how the virus look like. Or sometimes you want to precipitate the antigen, you can use this technique whereby we mix our sample with the antibodies. The antibodies will aggregate all the antigen, and then you can really see. And of course, the other one is immunofluorescer. You can use a different type of dye to observe a different type of interest protein in your sample. Okay, this is the facilities known as scanning electron microscope. We have one in IBS. Maybe this model, maybe a latest one or the older one, but this is how the facilities look like. Okay, it's very expensive. Somehow in one of these places it's been linked to the vacuum pump. Okay, and they are very expensive equipment. Okay. Okay, what's the definition of SEM? Scanning, electron microscopy. The name scanning also, it's just a scanning. It's a type of electron microscope that produce images or sample by scanning the surfaces. So you just look at the surface. For example, here, this example of a trachea or lung of a patient, just to show how is the antigen localized on the cell, or no, sorry, on the tissues. Okay, this is lung, must be the cilia, and this maybe this is just the mucus, and it's all the particle. Okay, so this is example of a surface of a lung infected by COVID-19. Okay, I use a lot of example of COVID-19. Okay, so that you're aware that it's an issue now. At least you can relate to what is happening at the moment. Okay, this another picture to show how people study the progress of the diseases. This is 4 day, day PR means that 4 days after infection, 4 day post inoculation. This is 24-day post inoculation. Okay, this is the terminate. This is tracker. They want to see the changes. Okay, here you can see terminate after 4 days look like this, and then 34 days look like this. And this is, as I mentioned to you just now, this needs a skilled people to interpret the changes. It's not easy, okay, because you have to know what does it mean by all this vacuole, okay, and changes from this to this. So of course they will compare with the normal one. And it's a trachea. 4 days, it look a lot of trachea, a lot of villi, okay, but here looks like the trachea is shortened, something like that, okay. But this is changes, of course, from 4 days to 24 days post-infection, post-inoculation. Okay, this study showed the co-localization of the MERS, okay, and the receptor probably in the respiratory tract and the lymphatic tissues of animals. Okay? This is scanning electron microscopy, just looking at the superficial, the surface of the organ. Okay, this listed also the advantage and disadvantage of SEM. Okay, give the advantage is 3D, topographical imaging. The instrument can work very fast, and you can save your result in digital form. Okay, and you require more sample, require minimal prep. Okay, so this advantage and disadvantage. Similarly, like a TM, it's very expensive and very large. You need someone to go for a training in order to operate the machine. If you know good, you're going to have a lot of artifact. Okay, it looks like abnormality, but it's actually abnormal because someone is not skillful to prepare the sample. And then sample are limited to solid samples. Okay, some also carry this radiation. This is common when high-tech equipment always using X-ray, and one had to be very careful when you are operating this equipment. Okay, this is TM. Okay, TMM refer that transmission electron microscopy. This is how the equipment look like, and we have one in Institute Bioscience. And if you want to have a go, you can have a look on the machine look like. It's very huge compared to your ordinary inverted microscope or the light microscope that have been used before. Okay, so here is the it says that TM is a microscopy technique. Okay, where the electron is transmitted through a specimen to form an image, and the specimen had to be in the form of ultra-thin section of less than 100 nanometer thickness. Okay, and then you can see the sample here. This is a sample of a cell. Okay, this here is the outline of the plasma membrane, and you can see here all these particle or the viruses is lining along the membrane. They are either attaching, wanting to enter the cell, or they are actually being released from the cell by cytoplasm. We don't know, okay, unless you have further sample. But just to show you that the virus is attaching to this membrane. And if you cut further, you can see that the location of the virus particle in the organelle. This you can see here in the vacuole here in the membrane, or the virus either they are budding, okay, or they are being going to release. We wouldn't able to tell you. Okay, this is coronavirus in the organelle of the cell. Okay, so the keyword here is that TM using ultra-thin section. You still have to cut the sample. It's just that your sample is going to be very, very thin. Comparing to the histo section that you normally see with HAE staining, that is about 3 micrometer thickness. But this is very thin, about 80 to 100 nanometer thickness, very, very thin. Okay, so that you can see a lot of changes. If too thick, there's so many things that obscure the important thing. But when section is very thin, you can really see the significant things that you wanted to see. Okay, TM also, you can, this is another picture, a large one, just to show you under TM, you can see how the virus is present in the organelle or in the membrane vesicle of a cell. Okay, the location, and you can see here, this is all the virus particle. Okay, I won't know whether they are matured or not matured. Uh, but it's just that this is how they will look like if you have your sample like inclusion bodies. This is how the virus look like under the EM. Okay, they are actually aggregation, accumulation of a lot of progenies of the virus. Okay, this all progeny after the replication of the virus in the cells. Okay, the next topic on the visualization of individual virus particle. Okay, I get this thing from the net. You can always check for the notes. Okay, let me read here. Most biological materials show little contrast with their surrounding unless they are stained. In the case of light microscopy, contrast can be enhanced by using colored stain. Okay, I think you have experienced examine histosection. Okay, histosection, you examine them under the lab microscopy and they are stained with H&E, hematocin and eosin staining, right? Okay, similarly with EM, it has to be stained. Okay, to heighten this contrast between viruses and background, use is made of electron dense stain. They are usually compound of heavy metal of high atomic number that serve to scatter the electron from region covered with the stain. If virus particles are coated with a stain, the fine detail may be obscure. So what is the intervention for that? So instead of using a positive stain, for us to study the virus particle, we have to use a negative stain. Okay, so negative stain overcome this problem by staining the background and leaving the virus relatively untouched. The negative stain is molded around the virus particle, outline its structure, and is able to penetrate between small projections, okay, and to delineate them. If there are cavities within the virus particle, they are accessible to the stain. This will reveal and some of the internal structure of virus might be disclosed. Okay, so theoretically, this is your virus particle, and you put this is stain, and when the light comes here, the heavy metal, you'll be to give an outline of like this. Okay, it will just stain outside. Okay, okay, that's how you can see here. This is coronavirus. All the stain is only stain, not stain inside the cells, not staining inside the virus, but staining outside the virus in the background, so that you can see this is a spherical virus and it has the envelope characterized by spikes or papilloma around the viruses. Okay, and that's how the viruses got named, because the spike looks very beautiful. They're just like a crown. Okay, so that's how the virus got named coronavirus. Okay, this is pox virus. They describe it as a virus like a brick shape. Okay, this pox virus is very big. Okay, sometimes the one is so big, maybe you can also observe under the light microscope sometimes. Okay, it's about 500 nanometer. It's very big. And this is another virus. Can you see this is speaker virus, and there are a lot of dented area here, do like cups. Okay, that's why in Latin, calici being cups. Okay, so calici virus is actually referring to the virus morphology with a lot of depression at cups, and it's how the virus got its name. Okay, and interestingly, this is a bullet-shaped virus. It's only one family, the bullet-shaped virus, that's a rhabdovirus or rabies virus. Okay, so you should remember this very easily because the virus has a bullet shape. The bullet shape, rhabdo means bullet. Okay, in Latin, so how the virus got its name. Okay, so this is example how one can appreciate the significance of EM in the diagnosis of viral diseases. Okay, I also have a videos for you. Please do your self-centered learning after this. There are 3 videos. The first video just to show you there are so many type of stain heavy metal stain that could be used in staining TAM. Okay, some with the advantage and disadvantage. For information, in UPM, this is very common heavy metal that we use for stain that known as phosphotungstic acid. But in this video, I think there are 6 different varieties of staining that you can see what the differences on the outcome of the staining. Okay, watch this video. And the second video is a simple method of how you're going to prepare a sample for negative staining. And the samples obviously is actually a supernatant or a liquid containing the virus particle. Okay. For example, you have your example here is that, for example, you have your cell culture plus positive with a virus where there is CPE. Okay. And then example here, you have your tissue culture plus, okay, and they show CPE, and you want to confirm the virus. What you do, the supernatant here, you will pellet supernatant, you will undergo centrifuge, you will pellet the sample, okay. How we pellet the sample? We put in the centrifuge machine, okay. And then this pellet, then you will take a drop of this one, and this was subjected into EM, negative staining, sorry, for negative staining. And the video show how, okay, negative staining, okay. And the video show you how you take a drop of negative staining, you wash them, and then you stain them with this heavy metal stain, okay, and then subjected to EM. Okay, after stain here, of course, you go to EM. Okay, the third video here show you how you want to examine a tissue section, organ which is infected, and from the organ you take the sample, you process the tissue, and you subjected for the EM. Okay, and of course, after watching the video, I want you to go through all these questions and answer them. Some of the answer is available during my lectures. Some of the answer is present in the video. Okay, what's the function of carbon grid in negative staining procedure? These different type of staining reagents in staining the carbon grid. Name a specific microscopy to examine negative staining. List the advantages of using EM. List the limitation of using EM. Why chemical and reagent reactor disposal need to dispose properly? Okay, why we can't just throw them in the ordinary dustbin? Why we have to have a special way of disposing this chemical? Okay, and for the second video, name 2 electron microscopy procedure they are commonly used to diagnose the viral diseases. Okay, if you pay attention to my briefing just now, you might be able to answer this question. Okay, the third video specifically to tissue processing for EM. Okay, there are a few questions. What is the thickness of the thin section? Okay, in this procedure compared to histology section. What is the purpose of performing TM on tissues of organ? Justify why the procedure of EM is very costly. It's not cheap. It's very costly. Okay, why? What is a microtome? Okay, you know this answer by watching this video. Why the carbon grid is used to collect the sample in this procedure rather than glass slide like other ordinary histosections. Why uranic acetate is preferred staining reagent in this procedure. And finally state the TM magnification times, okay, to observe the outline of your sample, the cell structure, and the plasma membrane respectively. Okay, this all. You can watch the video, and you can able to answer the question. Okay, this is some very general comparison of advantage and disadvantage of light and electron microscope. Light microscope is just a microscope that you normally use to examine histosection. That's light microscope. Electron microscope, that's what we have been discussing today. So in electron microscope, how much you change your objective, you can able to verify up to 1,500 times only, okay? And the depth of the field is very restricted, and this can be counter by using an EM, where EM you can magnify your view to 500,000 times. Okay, and of course you can possibly investigate greater depth into what happening in the cells. Okay, of the infected. Okay, this is very common. This is what we call light microscope. Okay, the light microscope also refer to optical microscope. It's a type of microscope that commonly use visible light and lens, using lens to generate magnified images. It's very common. You can see this type of samples. Okay, they are being stained with H&E. Okay, that's why you can see. Otherwise, they are colorless. Okay, so hematocele and eosine. Okay, so in the infected samples, then you can see this abnormality. This is what, if you describe here, this is all the nucleus, aggregation of the nucleus here. Okay, this is what called syncytium, CPE, and the CPE is characterized by the aggregation of the nucleus. Okay, known as also as a giant cell. Okay, because they only contain one single cytoplasm, but there's so many nucleus here, or also known as multinucleated cell, giant cell, or also known as syncytium cell. They are similar. Okay, another microscope, maybe you have used this in your pathology class. Okay, this stereo microscope used to dissecting things. Okay, the stereo microscope is often used on solid specimen to carry out close work, such as doing dissection, or you can in forensic engineering and entomology, study of the insects. Okay, this is the inverted microscope that the virology use very much to study the cell changes. Okay, this is what we call inverted microscope because the light all come from the top. Okay, and we have the lens at the bottom here. Okay, the definition is that this microscope is very useful microscope for observing living cells. Okay, or organism at the bottom of a large container, a tissue culture flask. Okay, and sometimes also in the heterogenealogy lab, they use it for manipulation application. And for the virology work, we use to monitoring CPE. For example, this is plus you put here, and you're going to monitor whether the enucleation you performed previously had caused any changes into the cell. This is look very normal. All the cells are intact and they are single cells, monolayer. Over here, you can see some of the cell is not normal. Okay, there's some space here, and some of the cell here bigger than the rest. And towards the end, after a few days, you can see that almost all of the cells here are infected. They are all become ballooning. They all become aggregation because they are filled up with the virus progeny. Okay, where is the virus? If the cell is being destroyed and lies, all the virus is let free. They are all floating in the supernatant. Okay, so eventually all these cells will die. Okay, another microscope. Okay, these are the cells, the picture that I normally use in the class to show you fluorescent antibody test. This is very common test, very specific because we're using both antigen and antibodies. Okay, and the signal can be easily seen. Okay, you can see all this fluorescent. This picture show that the fluorescent is present in the cytoplasm. Okay, so indicating that this virus is replicating in the cytoplasm of the infected cells. Okay, and rule by thumb, those replicating in the cytoplasm are actually they are mainly all the RNA viruses. Okay, areas which are not stained, meaning there is no virus and they appear dark. Okay, this is a fluorescent microscope. It's quite expensive. We have to maintain the UV light, and the bulb also is very expensive. Okay, and this should be handled with care. I think in UPM I think we have about 2 or 3 microscope only. Okay, in the lab. Okay, in certain lab. Okay, I've done with the. I think today's practical is very short. I have done with all my explanation. Okay, since we don't have any hands-on, you have to rely on the video that I show you just now for you to appreciate how we're going to do the technique. Okay. So if you have any question, you can please put in the chat box so I can see and try to answer after I finish with the last part. Okay, let me check whether there's any question. Okay, so far there's no question. Yes. Yeah, sure. You can open your mic.
Okay, hi Prof.
Hi, Mira.
Prof.
Is it?
Yes, Mira.
Yeah.
I think I missed it. Yang the slide, the 12th slide kan?
Yes.
The slide on the 12th page kan, there's a picture of negative staining uses TEM, and Prof mentioned that virus, the left side, the left picture, Prof mentioned that if you use a negative staining, you can only see the outline of the virus, right?
Yes. Yeah.
Oh yeah, I just wanted to ask, like, is it for the right picture? Is it the same?
Is it this one that I'm showing now?
No. The one yang the colored picture.
The color picture. This one. What's the title of the slide?
Negative staining uses TEM. In the slide just now. Tadi Prof yang yang ada
This one. Which one?
the 12 slide yang ada ditulis negative staining uses TEM.
Let me see.
The one yang Prof tunjuk tadi tu, Prof. Betul, Prof.
Yang tadi Prof tunjuk tu.
Tadi yang mana? Slide mana? Which one? Because,
Yang the first one yang Prof
okay,
tunjuk.
let me go through. Okay, which one?
Tak. Alamak. Okay, bukan. Okay, next. Next. That one, Prof.
Okay. Yes, okay.
Okay. Kan yang the colored picture, right? So
Where's the color picture? Where's the color one?
tu yang mana yang green tu?
You want the green? Ni, ya, rabies virus ni.
atas, Prof.
There's no color here. These all stained. They're all black.
Bukan Prof. Yang
Illustration, Prof.
I'm super explaining.
I don't know. The color picture is that's not TM. That is light microscope.
Huh? Yeah.
Let me see. This one?
Bukan Prof. Mira referred to the picture yang atas tu.
Yang atas tu, yang warna green tu.
Picture warna green.
Tadi tu betul, Prof.
This one.
Yeah, that one betul. Yes, that one.
This picture, this diagram.
That diagram.
Okay. So what's the question?
Tak, kan the left side. I think I missed the left side. Prof kata yang if you use negative staining, you can only see the outline of the virus, right?
Okay.
And then the right side pun sama
Yeah. For negative staining is there is only the staining is only the background only, not the virus. It's only the background.
Okay. Tu je sebenarnya, Prof. Okay, thank you.
Itu saja. Okay. We still need the background. Okay. There's no color here. Okay. The diagram with color. This is just to show you, okay, how the transmission of electron, okay, passing from the top to the bottom here. Okay, it's actually staining only the outline, not inside the virus. Okay, only the outline. Okay. So, any more question? Okay, there's no question. I will proceed with your next week practical exam test. Okay, I'm telling you the format is MCQ. You just choose one best answer. Okay, this is example. Okay, you will receive a question something like this. Okay, for example, the question is asking here The name rotavirus is derived from the virus morphology of, okay, the mark for this question is only one. Okay, for example, you answer the question, the answer is A. Okay, for example, let's say you choose the answer B, so there's no mark for this one, and I don't proceed for the justification. I will only proceed to the second step of the answer if your answer is correct. For example, if you answer number one here, the name of rotavirus is derived from the virus morphology of, okay, wheel, so you have to give justification. Okay, and the justification had to be very, very simple, short, and concise. Okay, I don't want you to write a lot. Okay, only one line. Okay, keutamaan, priority only one line because we're not going to go for the second line. So you better focus only one line, a simple one, something like this. Okay, the answer here, rota is a Latin word, means wheel. Okay, now second question, example. Of course, your question tomorrow, next week, be practical based. Okay, this is theory. Okay, for example, this is a question for the retroviruses. Avian leukocytes virus of subgroup J induces mother leukocytes forming tumors in. Okay, the answer is bone marrow. Okay, sorry, the answer is C, bone marrow. Yeah, C, bone marrow. Okay, so if you answer C, you got 1 mark. And then I'm going to check whether your justification is correct. Okay, I want to make sure that you understand why you choose C. It's not because you just guess. Okay, so I will see whether the justification you give is correct. For example, the answer will be something like myeloid. Why you choose bone marrow? Because myeloid is tumor of blood forming cell of the bone marrow. Okay, yeah. And the third example here. Okay, this third example here. Third, envelope viruses are relatively sensitive compared to non-envelope viruses owing to their, okay, the answer is C, okay, because of their envelope. Okay, why you choose envelope? Okay, this is the expected question answer I expected from you. The envelope originated from the host plasma membrane. Okay, so this type of a question that you're going to receive on Monday for your practical as well as for your final exam. Okay, your final exam also, there will be 60 questions and it's going to be in the form MCQ with justification. Okay, so next week will be a training for you on how you're going to answer this type of question, because we want you to understand really why you choose this question. Okay, but for next week practical, in addition to answer this MCQ question, you're going to, I think I have briefed you all before, we're going to do in the format or style of using IRAD and GRAD. Okay, briefly, when you first ready to come to the test, you should register in the Putra Blast. At the same time, you register for this practical class, and then after register, I want you to get ready to answer a set of MCQ question, 20 question individually by Google Form. Okay, and then you will submit online. Okay, and then after you've done that at 12:45, immediately after you've done this one, I want you to start to sign in into WebEx, and please register like how you registered today. Okay, don't forget your group number. Okay, your member, your name, and then your Patrick number, because you're going to make you into a group using channel application in the WebEx. Okay, from here, you will also receive the same question. You discuss among yourself. Okay, and then you come up with one answer. Okay, I hope that by doing this one, you will have discussion among yourself first. Okay, and then after at 3 o'clock, a leader which had already been identified will submit the question online, and then I will receive them, and then we will meet together at 3:30 to 3:45. You will all gathered all together in the main hall of WebEx room, and from there I will give a reflection. We're going to go through one by one question and see whether you got the answer correct, or if you don't get the answer correct, what's the reason why you don't get the answer correct. Most of the time, the student resolve all the problem during the GRAD, okay, group, okay. The student will peer group discussion. You almost of you resolve all the problem at this level. Okay, so if you have any question on this one, you can just ask me again during our lecture before the class or before the exam. Okay, the marking scheme, your practical examination conferences of only 10 marks. Okay, we allocated 100% for IRAD individuals. And then when you do your group discussion with the same question, you go on with the members also 100. And for this 10%, you will get the average marks from the IRAD and GRAD that contribute to your final exam or final marks of the 10%. Okay. Yes. You have question, Avishini? Okay, is there any question? You get on the mic.
Prof,
Yes.
I have a question. For the GRAD, we have to write justification also.
Yes. So in
Meaning that-
GRAD, meaning that you write justification, but after discussion with the group members. Of course, I can understand that where you sitting 6 of you, you got the same answer, but different people will have different justification. But I want you to agree on one justification. You have to explain. If someone got a different justification than yours, then you have to say out why you think that one, and you have to convince your friends. Okay, of course, your friend will also say your opinion. There's a form like a discussion. So only one justification submitted by the group leader. Okay, any more questions?
All right, Prof. Thank you.
Yeah. Is there any more question? Of course, group leader has to be responsible. You have to let me know who is participating. Maybe I will go around, okay, during that few short minutes, I will try to go around jumping from one group to one group to see whether this discussion is going on, okay? So the system able to make one person to jump around to see, okay? So I can just listen to what you have discussed, okay? Any more? Okay, no question? So you're very clear about this exercise? Okay, if you got any problem, you can ask me again because I'm going to see you again on Wednesday, okay, for our practical, for our lecture class. Okay, if there's no question, I think that's all for today. And you have plenty of time this afternoon. Please use this opportunity this time to go through the 3 videos that I have uploaded. Okay, the link is in the Putra Blast, and then go through all the questions and answer them. Okay, there's no question. Okay, if there's no question, that's all for today. So assalamualaikum warahmatullahi wabarakatuh.